Hello, everybody. My name is Joshua Ramon, and today I would like to talk to you about the exploration of tunable anti-cancer metallodrugs. Photodynamic therapy, or PDT, is a complementary approach to conventional forms of cancer treatment, such as surgery, radiation, and chemotherapy. In its simplest definition, PDT employs the use of a non-toxic photosensitizer that is inactivated by a specific wavelength of light to destroy cancer cells via singlet oxygen or another reactive molecular species. Currently, there's only one FDA-approved drug for cancer PDT, which is photofrin, a porphyrin-based organic photosensitizer. Second and third generation photosensitizers are also porphyrin or tetrapyrrolic-based systems. Our group in particular has had a longstanding interest in utilizing highly tunable metallodrug photosynthesizers for PDT because they have a variety of excited state configurations, which can be accessed at lower energy light wavelengths. Specifically, ruthenium and osmium-2 complexes are showing promise, with our own TLD 1433 undergoing phase 2 human clinical trials for non-muscle invasive bladder cancer. In this work, I'd like to present the synthesis and characterization of a Ru2 terpyridine-based family of photosensitizers and highlight some of their photobiological properties. There were two objectives going into this project. The first, to synthesize, characterize, and evaluate a new class of Ru2 terpyridine oligothiopine complexes. The second, to determine the effect of oligothiopine chain length and coligand identity on biological activity. Here we have our two target structures. The first one being a Ru-bis terpyridine complex with ruthenium as the central atom, and the terpyridine coordinating on either side. The second is very similar, with ruthenium as the central atom, terpyridine on either side coordination, but it also has the addition of this thiophene group, ranging from 1 to 4 in length. Moving to this middle section, we have the synthesis, characterization, and purification of our compounds. So both of the synthesis procedures started with very similar precursors, rutris chloride and our terpy or terpyridine source. In order to get this Ru-bis terpyridine, we use microwave irradiation for 30 minutes in a 1:2 ratio. In order to get our other compound, we started with a very similar 1:1 ratio, rutris chloride and terpy, but after 15 minutes, introduced this terpy NT coligand. This was the source of our thiophene. After the synthesis, the resulting chloride salts were purified using silica gel flash chromatography, and then to remove residual mixtures of nitrate and chloride salts, all the compounds were converted to their corresponding PF6 salts upon the addition of aqueous KPF6 solution. Anion metathesis was then performed on amberlite resin using ion exchange chromatography. Finally, the chloride salts were further purified using Sky's exclusion chromatography, and HPLC purity of greater than 95% was confirmed. Here we have the correlational spectroscopy NMR of our Ru4T compound and our proton NMR of our Ru-bis terpyridine and Ru-terpy 1 through 4T respectively. Moving to the far right-hand column, we have our photobiological activity graphs, and although there's a lot to look at on these graphs, I want to highlight a very specific area. Here we have a ruthenium 2T complex showing increased potency as compared to the other complexes in the family. This is odd because our group usually sees increasing potency with increasing thiophene chain length, meaning Ru4T is greater than Ru3T, then Ru2T, and so on. So current second batch of testing is underway to confirm these results. In conclusion, a new family of Ru2 terpyridine complexes were synthesized, characterized, and evaluated for photobiological efficacy against melanoma cancer cells, and Ru2T showed the highest potency in normoxia compared to other complexes in the family. We would like to thank the Department of Chemistry and Biochemistry at the University of Texas at Arlington. We would also like to thank NIH, the National Cancer Institute, and the National Science Foundation. Thank you.